1D, 2D, 3D, 3D coordinates, distance maps, strings. We identify similarities of strings, yes?
You said in the previous lecture that in terms of information content, the secondary structures are 1D.
Yes.
Why can't you say the same about the tertiary structures, since we can represent them as this 2D distance?
So now we are getting into the details. So the question is, I said secondary structure is 1D because I can present it as a string. Now you could strictly argue that the 3D coordinates of a residue you can also present as a string. This is sort of a little more complex string. And I take that part in principle. But the point is, if you think about this object that you're presenting, now I forgot to ask the question, but whoever, if you take the way you present it, then the string is one list of numbers. It's really a string. 2D is always a matrix. And in 3D, you need another component. To localize a point in space, you need a third coordinate. And those are the 3 dimensions. 1D is the dimension of a string where it's all about you and your neighbor. You advance in a string. 2D is in a matrix. So you could construct in a machine a way of writing it that looks like a string. But in terms of the way you think about it, it is a matrix, 2D. There's an IJ, although you can concatenate the IJs. The object itself is the protein. The protein has a length from 1 to 99. So you can sort of concatenate that into one long string. But the object really is 99 long. And not 90 times 90, 99 times 99. Right? You can write it as a string. But the protein, the reality of a protein is a 99 reality. It has an amino acid. This is essentially the way you think about what does it take you to write in a computer, on a paper, and in reality, the entity protein. Okay? If you look at the sequence, what it takes you is really for every amino acid as a representative, every position in the protein I has one position in your string. And that way you can write a sequence, and that way you can write secondary structure. That way you cannot write contacts. A contact map, you have to essentially, again, take every one of these i's and then have a relation of every I to every j, which is a matrix structure. Although given, you're right, I can concatenate a matrix into a long string. That is correct. But the unit really is the size of the protein. The basic unit is the amino acid I. And for I in a string, I have exactly, for each I have one position. In a matrix here, I have n positions, the length of the protein. I have essentially a 2D structure. I have to have the I against all the others. In 3D, I have the I in space. Think about it. We identify similarities between proteins by comparing strings, sequences, and we use generic substitution matrices such as Blossom to compare strings. That's a pairwise comparison, string to string. Again, the simplest would be amino acid identity. The advanced model uses Blossom, biophysical similarity. And from that you can advance into going into profiles, PSSM, position-specific scoring matrices, that change this idea of having a scoring matrix that thinks about any L in the protein and gives a certain score for replacing that by an A, to specifically what's the probability of the first L to be replaced by an A, and what of the second L in the sequence. And that is the position-specific matrix, and you derive that from looking at a count of symbols at each position I that you see in the family. This allows sequence profile comparisons. They are much more sensitive and allow you to sort of find things that you from sequence cannot find. without making mistakes. The next advancement over that is profile-profile comparison, and all I said about those, they're a little more tricky. What the take-home message, again, is simple sequence comparisons work somewhere in the daylight zone. That is where pairwise works. If we want to get into the twilight zone, we need sequence profile comparisons. If we want to sort of get into the deeper part of the twilight zone, we need profile-profile comparisons, yes?
The image of the profile of stripes, it's the impression that it's stars, but it's a full matrix.
You mean this image here should have a different coloring, or?
No, is a profile, is it the full matrix over the full length of the
protein? Yes. The profile really is position specific, which implies that every position in the protein, since it's position specific, there must be a specific value for every position. So the length of the profile, in that sense, is exactly the length of the protein.
PSSM is the same as
Profile, correct. The word profile and PSSM is completely the same, means exactly the same, yes?
So if you compare profile to profile, do you only compare two sequence families?
Essentially what you do, so the moment you have a profile, you have mathematically you have a matrix, right? So when you do a profile-profile comparison, you do matrix to matrix comparison. What does it relate to on the level of the protein? Yes, you essentially then compare two families to each other. So profile-profile comparison is a sequence profile comparison. You can also think about comparing a sequence to a family and profile to families. Yes?
You said that the profile is as long as a protein, but it's as long as the family, right?
No, yeah. Oh, that's a very good point. I have to revise my statement. I said that the profile is as long as the protein. If the protein has 99 residues, the profile is 99 residues long times 20. And here is a correction. Since the profile represents the family, the profile is actually as long as the family representation. That is correct. It depends a little bit on how you build the family. I did not have time to really discuss this on Tuesday because of the way the day went, but there are different alignment methods, and some of those alignment methods are, let's say, protein-centric. That means the length of the profile is exactly the one of the query sequence. So you build the profile as a, let's say, slave to align best with respect to your query sequence. So meaning you don't put insertions and gaps into your query sequence, because for you as an experimentalist, as a bioinformatician, or forever, this one is the one that you want to know something about. You don't allow cuts in that one. So everyone you align to it has to somehow be subdued to this goal that you have. That would be a protein-centric view, and in the protein-centric view, the profile would be exactly the length of the protein. And there's a family-centric view where you say, well, I don't care really about the protein, I care about the family, and then you're totally right. There was another question here. Yeah.
So a profile is the same thing as a PSSM?
Yes, the profile is the same thing as a PSSM, and I apologize for confusing you with using the same thing sort of again and again with different words. One reason why I do that is because I don't have enough self-discipline. One reason really is to make you aware when you go into Wikipedia or somewhere else. People use different words for that. They mean the same thing. And PSSM, I said that the first time I used the term, that these are the same. I will usually use profile, simply because it's just easier for me to say it. That's the only reason. There was this same problem that some people had with residue and amino acid. That is slightly different. An amino acid is what we think about the amino acid in isolation. The moment they are strung up in proteins, they're called residues. And bear with me, I try to avoid jargon as much as I can. So this is clearly not meant to torture you. Any other question? Okay, so that brings us to this issue of, can we predict 3D structures from first principles? I said that in principle you take this amino acid chain, you take the protein, you throw it into the solvent, water, and it adopts a unique three-dimensional structure. And we know that thanks to Christian Antonsen. He had the first experiments for which he got the Nobel Prize in Chemistry in 1972 that showed exactly that you can fold the protein, so make it adopt its unique three-dimensional structure. That's what folding means. And you can do it again and again and again. That's exactly what he showed. Now, if that is the case, then in principle, by understanding it's just water and the biophysical features of this protein chain that explain how it folds. I should be able to do that in a computer, in principle. Okay? by first principles. And not only, in fact, is it true that it seems that way, we have a variety of newspaper articles that stated that this problem has been solved. The first such paper was published in the 60s, second in the 70s, and the third one in the 90s. Now, it may sort of, some people immediately laughed. If you solve the same problem 3 times, then you see that something is wrong. And the third of these publications, and they were always, when science is in New York Times, then it means that something is really important that happened. So this is an important problem that people have been working on for a long time. So that brings us to the question, how could we actually assess? So these, what I can tell you, these newspaper articles were not because some journalists had not understood what has been published in scientific journals correctly. What had been published in scientific journals was completely understood correctly by the journalist, but the scientists had been, let's say, slightly over-optimistic about their achievements. Put it this way. Now, that brings us to this issue that it's actually not easy to assess whether you have solved the problem or not. How could we do it? How could we find out whether we really do the right thing? The problem why what went wrong is we did all the same thing that we all know from machine learning approaches. We have data, we take some of the data out, put it under the table and pretend that we don't see it. We develop our method. Once we have developed the method, we take this data again and see how well it works. This is what people do. But it goes wrong because actually the data is there, right? And people peek. So that is why a lot of machine learning goes wrong, and a lot of the following part of the lecture will be about how can we avoid these mistakes of peeking. How could you do that as a community? Can you think about a way to assess whether you can really predict structures? Yeah?
Have like a benchmark that only the maintainers know.
That's a great idea. So let me repeat what he says. I have a benchmark. So in this particular case, the benchmark implies a set of proteins for which I know the 3-dimensional structure that is such that only the maintainers of this data set know the benchmark. Great idea. Now the issue here is that each protein structure, so at the time we're talking in the 90s, a protein structure still costs about $1 million. Today it's about 100,000. So $1 million means you need, I don't know how many you need under your table, 20 million? That is quite some investment. So even in the US, for NIH, just pull out 20 million for a benchmark, they don't do that. Sounds like a good idea, companies don't do that. What else could you do? It's just not economically feasible, yeah?
Maybe.
I'm sorry, he was first, I'm sorry.
So in machine learning you have different splits and you train with one set and then you have one.
Yeah, but that's what I said, that's when people peak.
Jumble it around, so you have different.
Still, people peak. I mean, you're trying to what you do. We will again. I will talk about that in the following lectures, in lectures to come. But this cheating is not necessarily peaking. I'm using the word peaking because it's not cheating. You may in fact not even realize that you did peak. It's just, you looked at whether it's still there. You didn't actually look what it says. Hello. And it's amazing how we can make ourselves, we all know how easy it is to make ourselves believe the wrong thing. There was a point here?
Yeah, I would have just said that maybe you have to kind of train it on the data then by like benchmarking guide, but then you could still like fine tune it.
No, it's all not working. We need some hidden set that nobody knows somehow. This idea is the right thing, but the way you formulate it is just not economical, yeah?
Wait for new data?
Yes, so and that is in fact exactly what the idea of the CASP, the critical assessment of structure prediction, that was somehow triggered by the last of these problem has been solved kind of announcements, is, so the way it works is that there is a time in the year, so it's a biannual meeting. From April to May, the organizers collect experimental structures from experimental biologists that will be solved in the near future. At the point of the collection, the structure is not known. Okay, but we're talking to so I was at some point involved in this organization. So the experimental colleagues are asked, are you going to do a structure that will be published over the next 3 months? If so, please, can you contribute that to our experiment? So they collect the sequences, and then there is a period of prediction here. In the period of prediction where people can send in their prediction to these sequences, and the deadline is typically preset, because the experimentalist says we are going to determine the structure in August, so the deadline is August. And sometimes this happens faster. And then suddenly the deadline comes, in some cases, very suddenly. With the moment that it's submitted to as a manuscript was, that's the moment when it's no longer accessible. So it's like your idea. So we have something hidden without the economical price, because these things will be published anyway. And in fact, increasing even the incentive for experimentalists to contribute, because their structures will be visible in this context, right? So then all of these predictions, all of these structures are collected. There is a period here from September to November where the so-called assessors divine, as I call it here. And then there is a meeting in December where everybody sort of sees how well they did. This is an evolution of the first, it started in 1994, of the first years in terms of number of contributions and number of structures. But still, so initially, the one important issue is only homology modeling works. And that is what I'm going to explain what that is a little bit later. So it essentially means you predict 3D structure using similar proteins that have known experimental structure that look similar in sequence. I'll give you more detail about that. And the field is getting better and better in doing that. There is still no general prediction for 3D structure. So I cannot take every single protein in human and predict the structure. It has not happened over this scale. It has actually not happened until today. Yes?
But isn't this heavily biased against or towards easy protein?
This is heavily biased in some sense toward, so first of all, it's clearly biased toward the proteins for which we know experimentally the structure. And we know now that is a subset of proteins. You call them easy proteins, and that is totally right. So one kind of protein that is very difficult to do are membrane proteins. They're very important for pharmacy because 66% of all the drug targets are membrane proteins, and only 1% of all the known structures are membrane proteins because they are so tough. So yes, this is clearly biased in some sense. True. But still, that is not sort of the major issue. And another important finding is there are many improvements in many fields. One of the first big improvement was that directly after the first CASP meeting, people used each other's servers and built a lot of meta servers. And that somehow still dominates the field, although there are a bunch of sort of meta servers with their own name now. What you see here is over time, over time, this is target difficulty, means it's not quite the easy, you would call them, in some sense you could call them all easy, but in the easy ones, there are some more easy than others. These are more difficult, these are easier, and this is the quality of the model, so 100 means essentially is completely correctly modeled, and so the worst ones are not so well modeled, right? And this is 1994, and this is 10 years of CASP, and this is how the field moves ahead. That's the next 10 years, 20 years of CASP, and that's the last two. That was measured, so 3 years ago. You see that the field has improved rather dramatically fairly recently, and part of that, or some of that at least, is related to this announcement, and that is, in fact, from the last CASP 13 in December 2018, where a breakthrough seemingly happened by Google, AlphaFold, using deep learning to predict three-dimensional structures. And it's absolutely clear that this is the breakthrough that CASP has been waiting for a long time in many ways. So it's for the first time that a group with no expertise on the matter came to the top. And the other real point is that the way they do it is in many ways very different from what the other methods do. Although AlphaFold, by the way, at this point is still not published, we still don't know all the details about it, but it seems to be clear that there are many aspects that have been taken from other methods that succeeded in CASP. Here's another, that was a science publication, and that's a paper, data provided by Andre from UC Davis. So he's one of the people who maintained this experiment. And what it shows is the diagonal is an average. Above that diagonal, any point above the diagonal, every point here is one particular target. So another thing that you see, that a particular CASP event doesn't have thousands of proteins. In this particular case, it has sort of hundreds, a little bit over 100, right? So it's a little bit over 100 gray points. If the gray point is above the red line, that means AlphaFold contributed the best model to that one. There are more points above the line than below the line. That means that AlphaFold really is the best. But it's also true that there are other, so it's not that all the gray, that all the points are clearly above the line, right? So there's a variety of examples where AlphaFold was beaten by some other methods in the field, yes?
How you measure how good, or how you measure quality?
So what this shows essentially, how do you measure quality? So you essentially measure, at the end what you get is experimental three-dimensional structure. And for this experimental three-dimensional structure, you essentially measure how different, in something that is related to a root mean square deviation, how different is every single residue to the one that you predicted. It's not quite the RMSD, because the RMSD has a very peculiar feature about outliers, but it's some feature that relates to that. So how close is the predicted object, three-dimensional shape, to the one that you observed? It does not consider energy, it does not consider anything other than the shape. How close is the prediction to the experiment? Is that answering it? Yes?
I don't know really, but like maybe there are some parts in each protein that are more important in their structure than some other parts.
Yeah.
So maybe we know about this. You seem to say that we already know about this kind of knowledge. So is it taken into account in this?
So she asked, there are some regions and proteins that are more important than others. So one way of saying that is binding sites are more important. If I want to know how the protein functions, I need to know how it binds, and then ultimately the way the binding site looks is very important, somehow important for how it binds its substrate. Okay? Now the next question was the follow-up question. Is that taken into account by these methods? If available, yes. But for many of these cases, you do not have the binding sites. So for many of these cases, in fact, you need the three-dimensional structure to have any idea how the binding site looks. And then it's complicated to take into account. For some of the modeling procedures, so for AlphaFold, binding sites are not considered, or more important and less important is not considered explicitly. Whatever we know about the method, we know that they could not, in the relatively little time they had, put together something on that level of expertise. It is implicitly, because it uses alignment information, and alignments, again, it is most conserved where the most important parts are. So implicit, it is considered, but not explicit. Okay. Now, that gets me to another slight detour. I've said it before, I just want to say it in a more formal way. How do we actually get three-dimensional structures? These, in fact, are two ways of the cartoons. The way we do it by or the way it's done by experiments is by PDB has, and this is a little bit outdated, 2017, 120,000 proteins for which the three-dimensional structure was known. 109, so 90% of these were determined by X-ray, 9% by NMR, and 1% by EM. X-ray. So X-ray, what you do is you condense the protein so high that it actually forms a crystal. Some proteins, I said that think about cells as watches, not as a bag of water, but they're highly densely packed. And indeed, some proteins in the cell are in a state that is so dense that it's almost crystal-like. So for some proteins, this is very easy to do. You can essentially only scoop them up from the cell and get it. But these are exceptional. In most of the cases, this is not that easy to do. Once you have it in a crystal, and here's one example. When you see a talk from a crystallographer, they show you always these images. And for a crystallographer, this image is telling. So if you're a crystallographer, look at this crystal, and you know whether that's a good one or not. Some crystals look good and some don't. I have said in many of these, they look more shiny, or the image is sort of presented with more wow, and I know then I know this is a more important person, so when he shows a crystal, then wow, but other than that, for me, they all look alike. But again, these experts can really see the difference. So that image already tells you something about how good the structure will look. Now, you shoot X-ray beams, and I said you cannot just use your garden variety of X-ray on that. You need a synchrotron. You need a highly synchronized source. And in fact, many of the beams in the US today are mostly used for protein crystallography, for exactly that. And CERN as well, but CERN still has a lot of funding for other things. Now, then you get a scattering of everything that is in this crystal, and this scattering is such that where you see coordinated events, so crystal packing always at the same position, you have the corresponding atom, you get coordinated scattering. And that leads to phases. And these phases you fit into an electron density map. So I'll show you later a little bit what that means, this electron density map. Here's just sort of an illustration of an electron density map where you somehow see these dots, or pseudo see it, I'm sorry for the quality, and you have to fit the molecule into it. And then what you get out is an atomic model. It's modeled because you can sort of, into this electron density map, you can fit it sometimes slightly differently. In that sense, it's a model. But it's atomic. You essentially see every single atom. Well, some you may not see, but you see it on the level of the atom. Get to another technique, NMR. NMR has the big advantage over X-ray crystallography. You do not start with a crystal. You just take the protein in solvent, as it naturally occurs in the body, and that's sort of what this image here is supposed to convene, and put it into a magnet. Now, these again are not quite your garden variety of magnets. These are sort of capital machines. You see the person standing up here. This is not even a machine that is up to date today. Here you see a whole room of magnets. If you enter this room, it's a room that is bigger than this one here, much bigger than this one. You enter this room with a credit card, and you have no more problems about credit. Completely, you're wiped. So these magnets are immensely strong. And what you get out, what you actually measure, is the way the molecule in solution responds to the magnetization. And it falls back, it emits light from the falling back by the orientation of the magnet. That allows you to get constraints for the proximity of residues I and J. What makes it a little bit more complicated is you do not see constraints between I and J, but you see constraints between L and a V. But you do not know which L and which V, because there are several L's and several V's in each protein. And that makes it more tricky. And that makes it also very difficult to do this for very long proteins. The more L's in a protein you have, the more difficult it gets. So essentially this is sort of, you can do that to proteins that have 150 residues, 200 residues, but not much more than that. And what you get out here is you see there is in fact a bunch of structures. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and so forth. You see many. And you see that in some places of the protein, they all have a different place in 3D. In other places, such as this helix, they all fall together. And the interpretation of that is that those are alternative ways to fit your data, which essentially is a 2D map, and project it onto 3D. I said that 2D, the entire information is contained to get a 3D image out, but here we have mistakes in the measurements, or unclarities, or we have ambiguities, and they have to be resolved, and they can be resolved in an optimization process, and this optimization process has different solutions. That's why you get different solutions. And the interpretation of these different solutions is that you have dynamic ensemble of structures. That means that if you looked at the real protein in solution, sometimes it would look like this, sometimes it would look like this, and sometimes like that. Proteins are dynamic, they move. Where you have different traces here, where they differ, these different models, that is where there's a lot of motion. So here's a lot of motion, and there's very little motion here. So that in fact these different NMR models tell you about the dynamics. Okay, that's in many cases true, and that's the way to think about it. So yeah?
So NMR is kind of only works for small structures, but it's actually way better in kind of showing like the functionality of the structure because it shows the movement. Is it also cheaper?
So NMR has many advantages. I do not actually know whether it's cheaper. I do. Most likely it's not cheaper. So I believe that at this point, very often, like whenever you do things more often, then they get cheaper. And since 90% are done by crystallography, I assume that it is actually cheaper. There's a lot of expert work. So the X-ray structure can now be done by a PhD student today, in a sort of lucky case. NMR still cannot be done on that level. It still has to be a postdoc, so you still have to, so the way to get from the raw data to the final structure until you get to that image takes longer and more expertise. And that ultimately, if you penciled in this as to the price, then I assume that maybe they are somehow equal. If you only look at the machine price, again, it's difficult to compare. The synchrotron is way more expensive. But you're looking beam time, so you're renting some space. These magnets are protein specific. There's no big company that runs these magnets, and then some protein crystallographer comes and uses it. That's what they do in synchrotrons, so they're very difficult to compare these things. But again, these rooms that I show you here, for instance, that stands on 137th Street in New York, in Harlem. But this is an immense investment to just to prepare a room that is such that these machines can stand there. There's an immense amount of money that goes into that. That used to be, by the way, a gym. Anyway, it's in City College. Now, the other way in which you can see them is by cryo-electromicroscopy. Cryo, because essentially you freeze the protein first, because it's too dynamic to look at it like that under a microscope. So you have to freeze them. And then once you freeze them, you sort of have to work very well on your microscopes. And this has been one person who has developed that is Joachim Frank, who's actually going to speak at the IAS on the 6th of June, I believe, 2 o'clock, and got a Nobel Prize last year for this. Now, the downside to cryo-EM is the resolution. So what you get out from a cryo-EM can be on this level here, and you see the number 32 angstrom. Angstrom is 10 minus 10 nanometers. So this is essentially 0.1 nanometer. 32 is 3.2 nanometers, 16 angstrom, 8 angstrom, and 4 angstrom resolution. That essentially means how small is the object that you can really see. And what you see here, you see the protein as a blur. You see surface structures. You see that there are holes. You can look inside. But you're really not at all getting a position for every single atom. On this level here of 4 angstrom, you begin to, at least all the residues that are close to the surface, you can really see the residues and see them in 3D. Still, you cannot look inside because you only have the overview from a microscope. You could slice it, but then you would cut the fold, right? You cannot do that either. And cryo-EM mostly is working on somewhere in this realm here. In some cases, they really get higher resolution issues, but it is something that is up and coming, yes?
Can we crystallize and then slice it?
Could we crystallize and then slice it? So that, so far not. But I don't believe this is a response that is anything other than our technical lack of an ability. That maybe this will be the ultimate solution. This is also, by the way, from a paper here from the Baker lab, a way in which we can, there's another one from the Charlie lab. I thought that I had put this one up. Anyway, so you can also imagine that now you can combine computer modeling with cryo-EM, where cryo-EM gives you the background or the surface probability, and in the computer modeling you try to see how you could sort of fold inside, and you don't have the task of really folding a protein from scratch because you have sort of surface constraints. So certain things are possible, and maybe for some cases in fact that works. So a combination of folding in the computer and seeing the outside image. In some cases this is a successful strategy, and various labs are doing that. Now here I show you different resolutions of structures. So you may make it a little bit darker. It's still, I believe it's better now. The blue is, if you want, that's the electron density map. The blue is the experimental data, are the experimental data. At 3 angstrom, how you fit this ring into that, or it's simply better to see here. How you fit the ring is rather arbitrary, right? You immediately see that rotating that ring will not change the fit very much. You can rotate the ring and it will still fit to those experimental data. While in the case of this 1.1 here, there's only one way you can put this ring in. Completely decided, right? Now, the vast majority of the structures that we have are more on this side here. This is just 1.0 is just a handful, or a few handfuls at this point, of structures. So this is relatively few. So you can, on this level, really see every single detail, and there's no second possibility. On this level, that's why it's called model. There's still a lot of leeway, a lot of guessing. Okay. The only way, yes?
I'm not clear about what was happening in the previous slide. So there is like electron density map.
Yes.
And you're trying to fit a structure into that.
Yes.
And
so wait. So you scatter X-ray beams on a crystal, and what you actually get out of it after phasing is an electron density map. What you get out of it is the blue. What you know is underneath the structure that made this image. Now you know which atoms you have, and you just try to fit the atoms into what you see. Right?
yeah,
Okay. So that is 3D structure, but when I show you a 3D structure, I show you secondary structure. What is this thing about secondary structure? Well, I say it's 1D, but how is it, where on the structure is it actually? So there are 2 major secondary structure segments, or elements, secondary structures. One is the helix, one is the beta strand. The helix is, as you see, this object that sort of winds up, and the beta strands, 2 things come together. Again, I showed you this image, I showed you different ideas of showing the image, and the idea that secondary structure is in fact stabilized by hydrogen bonds that in the helix form between residue I and I+4. Okay? So every single residue in a helix has a hydrogen bond to one that is 4 away in terms of sequence, right? So if you wrote the sequence, residue 1, 2, 3, 4, 5. So one would have a hydrogen bond to this one. 6, here would be a hydrogen bond. 7, and so forth. Right? And as long as you have this hydrogen bond, you essentially wind up this helix. That stabilizes the helix. The beta strand is stabilized by hydrogen bonds between strands that come together. They could be from far away. And every other residue here has a hydrogen bond. Yes?
So these are all secondary structures?
Yes. There's only 2. So these are all secondary structures. There are only 2 of those, and the idea that they are formed by hydrogen bonds is the idea of Linus Pauling. In fact, Linus Pauling published a bunch of papers in PNAS, in the Proceedings of National Academy of Sciences, because if you're a member of the National Academy of Sciences, at least in those days, you had a free number of publications that you could have. You could publish as many papers in PNAS as you wanted as a member in those days, and he in fact used that and published, I believe, 10 papers and proposed 10 secondary structures, and they were all theoretical. He was a chemist and had some computational or model explanation why it would be helix, why it would be the strand, and he explained 10 secondary structures. 2 we have observed. That gave him the Nobel Prize. 2 are right. Well, if 2 in 10 are right and nobody else could have done it, then that deserves a Nobel Prize. That's what he got. And that brings us to this question. So he got the Nobel Prize for discovering or understanding how secondary structures form in 1954. When did they actually see the first structure? What do you believe? So somebody says, proteins form unique 3-dimensional structures, and there is a secondary structure. I predict they are helices, I predict they're beta strands. Gets a Nobel Prize in 54. When do you believe is the point that they see the first structure? Anybody throw a number?
Perhaps 10 years later.
Oh, that was a good guess. She said perhaps 10 years later. Remarkably accurate guess. So John Kendrew and Max Perutz, in fact Max had started it. Kendrew was essentially the student of Perutz and came out with the structure first because the one that Perutz had been working on for more than almost 20 years at that point just took longer to get the structure for. Hemoglobin was more complicated than myoglobin because in fact it has to form a dimer. Myoglobin doesn't, and that's why Kendrew got the structure faster. Now these were the first 2 structures, 58, 60, so almost 10 years after the Nobel Prize. So it is really remarkable to give a Nobel Prize for something that you haven't seen before, I find. But anyway, is there anything else that anybody sort of sees on this slide? These are the first 2 structures that existed historically. Do you notice anything at this time? You have seen many of these images. Is there anything you stumble over? Yeah? There's only helices. In fact, protein number 3 came in 67 and it also had only helices. And off went the idea that there in fact are 2 secondary structures, and people absolutely firmly believe there's only helices. Right? And then came the first beta strand. Okay, how do I assign secondary structure from 3D structures? Well, you essentially use this hydrogen bond by some energy calculation, and that ultimately is a method that is called DSSP. And I don't want to say more about it, you don't need to know more about it. You can assign secondary structure from 3D structures in a computer using essentially simple physics, based on the assumption that secondary structure formation is determined by hydrogen bonds. And that brings us to this issue of comparative modeling. As I said, Anfinsen showed that structures are uniquely formed on their own. Slides here come from Andrea Schafferhans. I'm going to show you a few slides how things change. So KRAS is a particular protein called an oncogene. Oncogene, some of you may know, that has to do with cancer. In fact, oncogenes are words that have historically been used for proteins related to cancer. That, in fact, is one of the first big candidates for a protein related to cancer, RAS. KRAS in particular. Now, this is KRAS. This is the way KRAS looks. You see these beta strands here, you see the alpha helices around it, and you see the binding site. This one here, in fact, is the mutation. G12C means that the original native protein has a G, the amino acid G at position 12, and that is substituted by a C. That substitution leads to cancer, or is associated with cancer. Now, what you see here, or what I show, are 2 different protein structures overlaid. One, the one that causes cancer, and the other one that doesn't. And you're actually not really seeing a difference between the two. Here is an example where you do see a difference. This loop here is slightly different between the two. But overall, you cannot even see that there are two, because they fall strictly on top of one another. This one amino acid change doesn't matter a lot for most of the protein. But in fact, you see the binding of the substrate is slightly changed. And this slight change in this particular case is the cause of a disease. Yes?
Is that the reason why it causes cancer, because it's so tough to spot for the immune system?
So now, the question is the reason why it causes cancer that it's so tough to spot by the immune system because it's so subtle? I cannot answer that question because we don't understand cancer well enough. So there are so many different ways to create cancerous situations. No, the reason, I would simply say, the reason why this causes cancer is because the minor difference is just adding up. This is such an important issue, that an important molecule, that the minor difference is adding so much that it leads to a macroscopic effect, and that macroscopic effect leads to a different growth of cells in this particular case. So it's ultimately about the processes that it triggers. Why is the immune system not recognizing it? Because it's a molecule from the cell. Yes?
But isn't it the case that if you have something like alcohol or other drugs, and they're very similar to other body-based substances, so that they react in the body. So is there any molecule that differs a lot and also reacts? Because I assume that two molecules or two like have to be the same to be in a way active in the body.
So is it true that if I look at two proteins that do the same thing, will they always look alike? Is that one way in which I could phrase a part of your question? Your question is more complex. Let me take that out as one sub part of your question. No. So this is in some sense this issue. I talked about the midnight zone. In the midnight zone I said many proteins have similar structures although they don't have sequence similarity. Equally, in fact, you can the most important part of a protein is the binding site. So all you need to do is sort of mimic another binding site. And a lot of the protein could look different as long as the way you hold this molecule, you present the molecule to the next protein is the same. So you can change a lot without actually changing function. And sometimes a single change has an effect. Okay? So two things that actually you look at and they don't look the same could have a similar binding site and could function similarly. By the way, drugs typically are small molecules. They are not the size of a protein, they're the size of the substrate here. So what drugs typically try to do, they sit in this binding place, and if the protein does something that you want to stop, then they prevent the protein from doing that. So in doing the simulation, they typically have to fit exactly. In that sense, you're totally right. You have to mimic the natural substrate. That's absolutely correct. But let me keep it there. This is, I'm making too much of a detour. I should not have used the word cancer. This is about showing that a protein with one amino acid change, in this we have a relatively dramatic case where it in fact has an effect, an important effect, but largely the structure is the same. What I'm showing here now, or what Andrea is showing here, is another protein, RASH. Now the lime, the light green and the green here have 85% sequence identity, so 15% of the amino acids are different, and still, in fact, in this case, both are human, but they still look very similar, even down to the level of the binding site. Okay, they are no longer identical. This is much more different than what I showed you before, but it's 15% that already changed, okay? Now we go further down. We go to a protein in fly. That, in this particular case, has only 28% sequence identity. So now we have actually changed 72. And I believe I can clearly pitch you the statement that these, all three, have similar 3D structures. Okay, again, you know, there are things where they're different, but there are a bunch of places where it's really difficult. Now you can really see that there are three. But, you know, they're very similar. We can even go on further here. in this particular case, we go down to the bacterial world, the purple protein that now has only 19% sequence identity, so 81% of the residues are different, and it still looks rather different. The wheel here is for that particular protein family. You see that most of these proteins are bacterial. Now, this brings us to this issue: what if experimentally I had the fly protein available, and I wondered what the structure of the human protein would look? What would you do? So say you wanted to predict KRAS or RAS or whatever, KRAS. You want to predict KRAS, but you don't have experimentally KRAS. What we do have is the RAB6 fly protein. What do you do to predict KRAS? What could you do? Again, I repeat, you cannot predict three-dimensional structure in the computer from scratch. You cannot take KRAS sequence, throw it into the computer and get its three-dimensional coordinates. What could you do? Yeah?
There's a certain difference between the one that we have and the one that we want. We can take a look at, there are a few other proteins in the database that have a similar difference and see how they compare each other.
So let me repeat that for everybody to hear. So we have some difference between the fly protein and the human protein, and I'm going to look at the database whether there is another pair of proteins that have a similar distance or difference. And then I do what? So essentially you take the fly protein, you subtract the expected difference, and then you get the human protein. You wanted to say something?
You could, you start with the fly protein, and you know where the different amino acids are compared to the human protein. So it's like little variations or perturbations that you introduce, and with a kind of more elaborated variational method, you could like shift a bit the structure step by step. And
so what she says is in some sense more sort of simplifying what you say, and in some sense making it more complicated. The way I understand this point here is you have you know the corresponding residues, and that is an important step here. So you have to first align these two. You need that for your method anyway, and you implicitly said it. I just want to make this explicit part. So you have the fly protein, the human protein, you align the strings. Okay? Whatever we talked about before alignment, that's the first step. Now you have the two strings aligned. Having these two strings aligned means that you know where the corresponding positions are. Look up the fly protein and you sort of model the corresponding human protein. And she said you in this modeling you successively introduce some sort of modeling rules. You didn't say that explicitly. I take it out of your mouth because I'm going to say something like that in a minute. And that's exactly the way it works. One way to think about it is you have this universe of protein structures that is available experimentally, and with your new sequence you try to map them to one of these, and if you can map it, you use that and you predict. So you begin with your sequence, you align them, and then ultimately once you have this, so how do I identify the template? So how do I identify which protein it fits to? That is again done through this alignment. You find something that is similar enough, and there's a variety of methods that you can use for that: PSI-BLAST, HH-Blitz, or more refined methods, profile comparisons, and then essentially you build the model. Building the model, essentially the simplest way of doing that, and given the time I will just leave it there. The simplest way of doing that is simply you take the coordinate from the fly protein and you copy it. You have the corresponding residues in the human protein, and you copy it for the corresponding residue, right? So you have so in the alignment, in the fly protein, you see that has an ACG, KL, L, or something like that, and the human has a G, C, A, R, I or something like that. That's human here, and that's fly. That is 3D known, and that is the unknown. So for this C, you know where it sits in 3D, right? Now you're just going to say this C sits exactly there. You know where that sits. You put the A on top of that position. If you do that, there is this issue, and that's exactly what you said, that you sometimes, you may not fit there. Because the amino acid that you put in, here this R, has a longer side chain than the K. So you don't have enough space. How do you do that? Well, you try to move it a little bit in the direction to create space such that you can fit the R into the coordinates of the fly protein, right? So that's some refinement that you have to do, and that gets to the point that if these two proteins are very similar, the major problem that you actually have is speed of doing all of this modeling. Then you go further away, so I'm talking about your sequence with respect to the protein for which you know the 3D structure, the human in this particular case, to the fly protein, right? So the further it goes, the more the quality of the modeling becomes a problem. When you go even further down, then the problem becomes the alignment. So if this initial alignment here is wrong, then you would build the wrong model. No way. Mistakes from the original alignment, yes, you can move things around, but to jump to the next coordinate point is complicated. Then you actually start modeling 3D again, and then we are at a level where it's not easy to do that. And the further down we go is really the problem of detecting the solution, meaning you do not find that these two things in fact are corresponding. State-of-the-art models, one is the Charlie Lab modeler server, and ultimately I'm showing in detail here what I just talked about. Let me just look at one aspect. Once you have these two things modeled, what if we had a loop, and that loop in the other protein is not there? So you have the core of the protein modeled, but one has an additional loop. This additional loop, say, are 5 residues. What could you do with these 5 residues? How could you model how these 5 residues, in your fly protein you don't have the 5 residues, so you can't look up their coordinates. Yeah?
You have the database of previous structures, so you can take a look at the 5 residues in order to not.
That's exactly what people do. Let me just repeat what he said. So, and true to what you said before, you look into the database and you find, in fact, 5 residue loops. You look at all the 5 residue loops in the database, and you look at the ones that are most relevant in terms of sequence or the way they look. You know where this constraint is, where you know where that constraint is. You know that you have to fit it to these two ends. And you find the loops that fit best to these two constraints. That's exactly what people do. For shorter loops, you can then go into the level of really doing molecular dynamics, so using force fields, so using computers to simulate the event of folding. Simulating the event of folding, I said we cannot fold proteins. Well, that is not quite true. So there's special hardware done by the Institute of David Shaw in New York. So for now, I believe more than a decade, he has built specialized, super big computers. The first of those was called Anton. I believe they're still called Anton, just that they're in the level Anton 3 or something like that. And they really look cool. So there's a lot of money in this. And they are the fastest way to do molecular dynamics simulations. They can simulate, predict how proteins fold, but only for relatively short proteins. They clearly can do it for these loops. Okay, for additional things that you have. And another story is the one that started with a server called Swiss Model. And ultimately the idea of Swiss Model was to do exactly what I said before, find the protein that is most similar and copy the coordinates, and today they do much more, and it's very, very similar to what the modeler from Charlie does. Those are the most used 2 programs. Now, When you get these models, one important thing is, so this is the image here from René Magritte. It says this, in French, it says this is not a pipe. What does it mean? Well, this is not a pipe, so this is not the translation I'm looking for, yes?
So this is actually an image of a pipe.
It's an image of a pipe, not the actual pipe, right? That's what René Magritte wants to say with that. And this is the same here for the model. When you have the model, Thorsten Schwede, oh no, this is not from Schwede, this is Bent, model must be wrong in some respects, right? So one way to approach a model is always to ask yourself, you know it's wrong, right? Otherwise it wouldn't be a model, otherwise it would be the real thing. So where is it wrong? And what Thorsten says, a model ultimately is exactly the thing that helps you explain the data and come up with designing a new experiment. That's what a model is good for. And a model in that sense is only as good as its usefulness for whatever you want to do with it. That's the message here. But homology modeling, let me put that down as an important point because I have so little time. We can model every other of these. I said we have about 150 million protein sequences that we know. And we have 120,000 structures. There's a big gap between those. Comparative modeling, as this technology is known, allows you to cover half of that. So around about 70 million or 75 million of the proteins for which we do not know the structure, we can model. Simply mapping them onto the 120,000 structures we know. Okay? So this is the most powerful, most accurate way to predict 3D structures, comparative modeling. Easy, works very well, is used every day. Yes?
Are such algorithms part of the CASP competition?
Yes, so the comparative modeling is part of CASP, and in fact the big advance since 1994 is that in these methods every single step that they do has been improved. So that is the biggest improvement. So essentially one way of saying that is the prediction of, and also the reason is that the more structures we know, the easier this gets, because the target or the one that is more similar is, they're more likely to be things that are more similar to your protein, filling up the space.